Hello, sir.
Yeah.
Sir, yesterday in the session, the night session, it was mentioned that we hold some kind of dark interest.
Sorry, Govind, I can't understand you. Can you repeat that?
Sir, can you hear me now?
Yeah, I can.
Well, yesterday it was mentioned that there was some in the night session that there will be some Darwin Finch's evolution kind of session held in the class today. Would that be held today?
No, 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 no. No,
It's on Tuesday, sir.
I don't know. I don't think so. But yeah, I'll ask Robin. But no, I mean, Robin didn't tell me. Was
Uh,
this told by Robin?
well, sir said that he, due to my after Maya and Lakshmi's doubts, he said that suggested that you or this team may hold some kind of evolution-based study questions based on Darwin's features and similar examples.
Oh, is it for week 10?
I think so, sir.
Oh, then this is week 9. So, yeah.
Right.
So it will be next week then. So we'll start in like about 1 more minute. All right. We are quite a few people now, so I'll start with the first slide. So before that, I'll introduce myself. I'm Jobin Varughese from IISER Tirupati, and I take these doubt clearing sessions. We've been doing it for the past 8 weeks, and this is the ninth week. And this is for the basic course in ornithology, run by a lot of professors who are studying bird different aspects of bird ecology and evolution across the country. So, and this is part of my PMRF fellowship. All right. So the introduction is done, and I'll start with the first question. This is for week 9. Yeah. So I want people to answer these questions in the chat box if possible, and then once a sizable number of people have answered, then we can discuss the answers. So, Vikas Pal Singh, why do you think it's D? Can you explain a bit? I mean, not if not exactly, then can you try and explain it a bit?
Okay, sir. Actually, I guess that number of days in a week, they may be observed. We can observe these in continuous way only 7 days in a week. So I think so. In each category, it cannot fall. That's simple. I guess that.
Oh, okay. All right. And Tarun, you said it's B. Why do you think it's B? I mean, if it was a guess, then it's all right, totally all right. But do you have like a supporting argument for that? As well as Roos Bay.
Should I take a stab?
Yeah, go on.
It's basically, even if it's continuous, that's, these are not numeric values or something.
Yeah.
So while you can grade them in a continuous range, I think it's ordinal.
So nominal and ordinal are not continuous, so they are categorical. That's for sure. So definitely C option doesn't sit over here because day of the week are not continuous exactly. So, but why B and why not A? Is my question.
Because you could give them an order just for the purposes of, so say, supposing you want to really grade it week long, results of something, you could show it from Monday to Sunday.
All right. So it depends on what we are.
Would depend, yes,
okay.
Of course.
Can we also say that day of the week is a cyclic data? People did go through the lecture, right? Suhel's lecture. They do know what cyclic data is.
Sir, I haven't watched week 10 yet.
Yeah, you can't come and can't come and answer and look for a shortcut. You have to go through these lectures. Actually, Suhel's lecture is one in. You guys were very insistent on me sharing some statistical papers and books, and I'm sorry I couldn't do that, but Suhail's lectures actually touch upon, those two lectures actually touch upon a lot about statistics, and once you're clear with that, I think you don't have to worry
I
about.
know. Well, I watched it last year, sir, but this time I had an exam in college, so I didn't have the time yet. I'm planning on watching this weekend.
Yes, Saptarshi Sahaj says that day of the week is cyclic data, so it can be said as cyclic, yes. So my answer, I mean, so yeah, but it can also be said as nominal. I mean, although my answer that I have marked over here is nominal, but like Ruspe says, it can be ordinal as well, and it can be cyclic as well. So it would depend on basically the, I mean, the question that you're asking, and yeah. So there is no exactly not a correct option, but if somebody asks me, like, I mean, randomly on the street, like if the day of the week is ordinal, then that definitely not. If it's continuous, then definitely not. If they say if it's cyclic, then yes, and if they say that it's nominal, then yeah. I mean, each day of the week doesn't exactly rank higher than the other, so yeah, it will mostly be nominal or cyclic data. Anyways, yeah. So for those of you who didn't understand what cyclic data is, basically, like you can have so you can have the direction, the direction of say the nest hole of in a tree. You can say that it either directs to north, west, east, and that can also be very specific to what the bird wants. Like if it's in a temperate region, probably the nest direction could be somewhere in a more sunny area or something like that for the warmth. I'm not sure of the data, but could be. So, but the nest direction, the direction like north, south, east, is actually a cyclic data because there is no zero to it. I mean, you can have the value as 0 to 360 degree, but once it becomes 360, it again starts from 0. So there is not exactly a 0 to it. It's just like, yeah. So that's a cyclic data. Day of the week can be a cyclic data. Year of the month. So currently we are we have like a 2 year sampling of bird distribution. So we are if I look from the day we started sampling, then that would be like I mean it will be like a linear data, like it will have the next year it will be 365, the next after that year next year would be 365 into 2. But basically if I have to look at the weather cycles and everything, the year data actually becomes cyclic again. So after a year, the same seasons start again. So it is kind of a cyclic data. So yeah. So A for question 2. So people think length of a bird tarsus or depth of a bird beak is a nominal or ordinal data. Yes, Rusbe is correct in the sense length of a bird tarsus is basically measured with it's measured with the caliper, vernier caliper. I mean, you must have seen a very fancy looking vernier caliper, especially for birds and all. So you must have seen a vernier caliper in your physics classes in school. So they measure around like two decimals, like the first decimal, the one decimal place. So it could measure like 23.5 something like that. So it's basically a continuous data. So 23.5 and 23.4 can still have like several values in between, like several as in millions of values in between, infinite values in between. So that is a continuous data. Even the depth of a bird beak is again calculated in millimeters, so again a continuous data. So it's not exact. Nominal and ordinal, to make it clear, are categorical variables. So there are categories. So if I am saying so the beak color of say Indian blackbird in Sri Lanka and in India. So in Sri Lanka there's a brighter shade of red, so we can call it a bright red, and in India it's a shade of yellow orange, so we could call it yellow orange. So basically the categories would be yellow orange and bright red. But how can that become a continuous variable? Beak color. Anyone wants to stab at the answer? I mean, stab at that question. Like, how can we convert a color parameter into continuous variable? People know Photoshop and such thing, right? People are using yeah, if you actually give color values. So there are other things also. Color, there are others like something called hue, saturation, tint. These are the values that each color can have, and so people don't usually give red, orange, and yellow, because that's a very I mean, it all depends on the observer. The observer might see an orange and call it a yellow orange, whereas another observer can call it a yellow orange and not orange. So it's very subjective. So to give it like a very objective, yeah, to give it a very objective look, to objectify it basically. So we have to convert the colors into values, and particularly in terms of its hue, saturation, and stuff like that. So there are like continuous, that again becomes continuous variable for each color. So that's how you do it, and yeah, and which is better, a continuous variable or a categorical variable in terms of the result that you get from the analysis? Which do you think is a better way of collecting data? Yes, so Roosevelt says continuous is more accurate and lends itself to analysis. Yes, I mean, one can still work with categorical variables, but the only problem is, yeah, I mean, the test used would be different. That's one thing. But the other thing is, continuous seem more accurate since it's more numerical. Yes, it is actually more accurate, and like a good scientist would actually recommend you to collect continuous data on the field. But sometimes because of logistics or something, I can divide a wind speed. Wind speed is important for bird detection, so I can sample my wind speed based as heavy, light, heavy, or very heavy, or something like that, or even cloud cover as sunny, partially cloudy, and overcast, or something like that. So those are categorical variables. But I can also I can also make them into continuous variables by actually measuring the wind speed for wind. By using there are instruments to measure wind speed.
Hi.
Yeah.
IISER, since unlike continuous variables, categorical variables are placed in different categories. So in order to get a true measurement of them, you need to compare them with the rest of the pieces in the category, right? They're unlike numerical continuous variables.
Yes. So I think what Govind is trying to say is, suppose we use categorical variable and we have three things that we are testing. So within those three things, within each category, you should have like a very significant sample sizes. So basically, suppose we are testing, I don't know, I mean, we are testing mm, like speed of elephant or something like that, the speed of elephants, so walking speed of elephants, and we are looking for categories like juvenile, male, female. So within each category we have to have samples, I mean large enough sample sizes to be able to say something. But if we give the age of the elephant, if we know the age of the elephant, if we give age of the elephant, juvenile and adult, let us take only two categories, not male female. Let us not do that. Adult and juvenile, we are trying to say, we are trying to study the speed of elephants. So within adult and within juvenile we need to have sizable sample sizes. But if we can convert this categorical variable into just numbers like age of the elephant, if we know how to, if we somehow know the ages of the variables, then we can have continuous variables for the elephant age and the speed. So then we can have we don't, I mean, we would require still a sizable size, but we don't need that many that many sample size as was required for categorical variables. So that's the problem with categorical variables. So anyways, next question. Yes, go ahead.
Sir?
Yeah.
Well, I have a question. The
Yeah.
different colors and or shades of a bird, would they be nominal or ordinal, sir?
They would be nominal, unless unless there's a range that you know, but still they will be nominal. Yeah.
But what about, say, the different shades of a color red? Wouldn't that be ordinal?
No, that would be continuous. No, if you can quantify the shades.
If it's not, if you don't quantify it, would it be ordinal?
Depends. Like, if you are saying, I mean, that also depends on the question that you're asking, right? If you are saying that
From light to dark?
yeah, something like that. If you are looking for that, then probably it might be ordinal if you can somehow rank them. But ranking colors is slightly very tricky unless you have ranking colors is very tricky. That's the problem. Yeah, anyways, people are saying A as nominal, but basically so if you look at Suhel's lecture, presence absence are usually given by 0 and 1, so it automatically becomes like an ordinal data. where 0 is less than 1, but it need not be like that. Like that, it could be male, female. So sometimes males are given as I mean, generally females are given 0 and males as 1, but there's no rank to it. It's just 0 and 1 as a nominal data and not an ordinal data. So it all depends on what variable you're studying and what kind of a study that you're conducting. So this could be nominal as well, but yeah. All right. Yeah. Okay, I think a lot of people know the answer for this. It is mean. So yeah, I don't think I have to explain over here. Hopefully people understand and go through Suhel's lecture. So if you had gone through Suhail's lecture, he explains how a population sample, how a sample can be divided into ranges. So basically, the middle part is called like uh, like a variable from a sample population. It has an extreme at both the ends, the lowest extreme, the least extreme, and the highest extreme. And if you divide it into two parts, basically two sets of data. So one represents, if you basically first of all you rank them based on the variables. So the highest, suppose you talk about as simple as something like marks of a person in a test. So you start from the lowest marks to the highest mark, and if there are 30 people, then the first 15 would have the least range, and the second 15 would have the highest range. So what is the middle part called? I mean, what is the middle generally called as in this case? Yeah, your options are mean, median, mode. Yeah, so Tarun says median. Yeah. So the middle is basically the median, and you have everything. No, it's not called middle, Govind. It's called median. So the
I guess that the median is the middle, right? So that's what I.
no, there's a technical term for it. But yeah, so median, the lower range is yeah. So basically median. So then if we if so that is basically 50% over here and 50% over here. We can similarly divide it into 25, 25, 25, 25. So 4 25%. I mean percentiles basically. So not percentile. So it's called a quartile. So quarter. So 4 quartiles we can divide. So the first quartile and the third quartile. So whatever comes between the first and the third quartile is called your is basically is covered. So that covers around 50% of the population. 50% 25% on this side, 25% on the higher side, and there's a 50% in the middle, and that range is called the interquartile range. It's called the interquartile range. So yeah, so similarly, if you convert the same data into, so you can convert that into 4 parts, that becomes a quartile. If you convert that into 100 parts, then that becomes what? You might have, you should be knowing this by now, because not because you heard Suhel's lecture, but also because it's a very, yeah, it's a very common, yeah, 100 parts is percentile. So people know this term. So yeah, I'll just move ahead. So when I say chart, it's basically if you're looking at your data in Excel or something. Oh. Sorry, I had a power outage again. Sorry, so did you guys give, you might have to give the answers again. I'm sorry. So basically, in an Excel sheet, if you're trying to look at a data set and trying to see which is the highest quartile, which is the lowest quartile, and where does the interquartile sit, then what? Yeah. All right. Yeah, I think the answer that I'm looking for is box plot, and over here you can see how a box plot can explain a lot of things. For example, the interquartile range, which is like the 25th percentile and the 7th percentile, what the range between that. Basically 50% of the population comes here. Then you have that tick. The tick is basically one Q1, the first quartile minus 1.5 interquartile range on both the sides, and anything that lies beyond that are outliers. So yeah, I hope people got the meaning of this chart on the screen. and yeah, so now box plot are uh, being, I mean, in the latest studies, people don't, I mean, they still use box plots, but there are different ways of converting, I mean, there are different ways of representing the same data in a similar way. So now people are using something called a violin plot, where wherever the data sits high, so you have wider like a wider uh, width, I mean, basically the abundance of the data points gives you the idea of how the data is sitting. So basically it could be like a yeah, it could be like a violin, so that's called a violin plot. You should look these up, like then there are notch plots of, I mean, like different plots that people have been using instead of a box plot. Yeah, but it's like a simple way of representing your data basically. uh, People have answered column plot in the last this thing, but I'm not sure if column charts can actually tell you the quartiles and the interquartile ranges of a data set. That's why I'm
What
not.
about pie? What about pie charts, sir?
Pie charts would definitely not tell you what that. First of all, pie charts have to be categorical. I mean, so within a category, these are the values, or these are the frequencies of yeah. So
In pie chart, generally we show the percentage.
what? Sorry.
In pie chart, generally we convert the percentage fraction of our data.
Yeah, exactly. So that is basically frequency. So basically, when I, okay, so how do I explain? So I am looking at yes, Dave, go ahead. Dave Ball, you have
I
the,
have to unmute. Yeah. Okay. I was thinking about the column charts.
yeah,
Now, if I put the 4 quartiles
yeah,
as 4 columns and put them side by side,
ha,
that's a sort of otherwise, say what you have in a normal distribution is a sort of a continuous curve.
yeah,
Now, if you just put these quartiles
ha.
in a column chart, that
How would you?
stack, put it as a stack column or something.
But you still would have to convert the data into quartiles before you put them in the column chart, right?
But I
Yes,
mean, identifying the column chart, I mean, a column chart is anyway used for the frequency distribution, the
Yes.
histogram, right?
Yeah.
So if I just identify with some marker or something, what are the quartiles, right? Then it still can be used for representing the data, right?
So you're saying you'll divide the data first into quartiles, basically the first 25. So basically, if you have a data, say for
right.
if you're dividing a continuous variable into a categorical variable and then putting them in column chart. Is that what you
Putting
say?
them in the column.
It could be either I mean continuous variable. I mean because this doesn't say whether the data is actually discrete or not, whatever, right?
Yeah.
So
Okay.
my question is, or maybe Dev's questions would be whether the column charts really does not fit it all or not.
Yeah, I mean,
So I mean I'm just okay. Let me just look at it. So if you have a data set and you want to look at the quartile and the interquartile range, so
Right.
definitely, how would you do that for a categorical variable? I mean, how would you do that for anything but continuous variable? Is what I'm thinking. Is there a possibility of doing quartile and interquartile range for for a non-continuous discrete variable?
yes, say something like marks obtained
That is
by
a that is a continuous variable.
by say 100 students.
Yeah, that's a continuous variable though, but something
Yeah.
else, some other example.
It's not a continuous.
It is
It's not
continuous, right?
discrete, right? Like
It's
for
not
each
discrete, yeah.
person.
Yeah, it's not discrete, but it's a continuous variable. The marks are continuous variables. You either get 0 or you get 100 and anything in between 0 and 100. Right?
Okay.
So
Right?
you're trying to say with whether the what do I say, distribution? I mean the population abundance, could that be a part of this data?
That's a count data, yes. Yes. So you're saying, okay, so Suhail hasn't touched upon it, but count data has a very different distribution. It doesn't have a normal distribution. So we have to be very careful with that. It's got a something called a Poisson distribution. I don't know if you've heard of it, but POISSON.
Yes.
Okay.
So
Where
usually all
mean
the
and standard deviations are equal. Like,
yes,
yeah.
yes, yes. The mean and the standard deviation are equal in Poisson distribution. So that is, yeah. I mean, we are going to a very different level right now. But yeah, I
Right.
mean,
No, it's
yeah.
fine. Probably we can go ahead. I mean, this is just
No,
a
no, no. The discussion is nice,
topic.
but I like it. But basically, if you are, yeah. Anyways, yeah, it's a very long process if you convert the continuous data into categorical variable and then do column charts. But yeah, that can be done as well, I suppose.
Yeah. Okay. Fine. If you manage to sample every individual of the population in individuals for a variable, what will be
No, so a lot of people said n minus one, but Astagmat said a n. Do you have an argument for that, Astagmat, Mister?
Yeah, when you have all the population counted, it will be n.
Yeah. So yeah, it was there in the lecture as well. So when you're just
Yeah.
doing sample, and you are finding the whatever mean calculating the mean, then the degree of freedom is usually because it's yeah. So then the degree of freedom is usually n minus one, but you have every individual of the sampled calculate. I mean, of the population sampled, like a probably a census, then the degree of individual becomes total n and not n minus 1 anymore. So yeah, so the answer is A. Degree of freedom. Yeah. That's not error, it's error, okay? The option B. Yeah, all right. So I think a lot of people have already got this answer. It's called bootstrapping. So yeah, that's the extent I can explain possibly bootstrapping. But yeah. So I'm adding or subtracting standard deviation from my sample mean on either side. Matlab basically, so if it's subtracting, then it's on my left side, and if adding, it's on my right side. Okay, this is for a general normal distribution curve. Yes, you are correct in assuming 1.96 is almost equal to 2. All right. So I think Dave has already given you a hint. So when I said 2 times, I was actually meaning 1.96, so nearly 2 times the standard deviation from the mean sample mean. So suppose I have a mean of 15 and my sample standard deviation is say 5. Then I am subtracting 2 times 5 from 15, so that will become 5 on this side, and adding 2 times 5 on this side, so that will become 25, I suppose. So between 5 and 25, the the sample that I had collected with a mean of 15, I am covering basically between 5 and 25, I'm covering basically 95% of the population.
Did you mean here 2 times the standard error?
Standard error is for the mean of means. Yes. No, I meant standard deviation.
Because the formula was covering 95% range when it was x mean plus minus 1.96 standard error.
No,
Not
the same is applicable for SD also. If you
SD,
just go
yeah.
through the.
Standard
Okay.
error is for mean of mean. Standard
Right.
deviation is for the sum. Yeah.
Okay.
All right. Okay, this might require a bit of mental, or I don't know, calculation over paper or something. But yeah. Okay, so we can. This requires a formula. Not exactly. Uh, okay, the formula is s upon root n is equal to standard dev. So s is the oh s upon root n. So s is the sample standard deviation, and s upon root n is equal to the population standard deviation. Okay, so we can divide this into two parts. First, find the standard deviation of, that we actually require, that we actually need to calculate. So, if 16 centimeter is the confidence interval range, given 95%, it's like 1.96 standard deviation on either side of the mean. Like from the last question, what we understood was the 95% of the sample gets covered with 2 standard deviation on either side of the mean. So if we are doing 16 centimeter as that range, then 16 centimeter divided by, okay, I've done it somewhere. Yeah, so 16 centimeter is 2 standard deviation on this side, 2 standard deviation on this side. So how many standard deviation, I mean, what would be the standard deviation of the population over here? 16 centimeters is the range. So there are
4.
Yeah, so there are 4 standard deviation within the 16 centimeter range. So that would mean the we are achieving at least we are trying to achieve at least a standard deviation of 4. Now I collected and now I collected sample from 10 nest and mean being 150 centimeter, and the standard deviation over here for the 10 nest is 24 centimeter. So basically the standard deviation of the sample population is 24 centimeter. So if the formula is sample standard deviation upon root of n is equal to population standard deviation, we are assuming both are same, and then what would be the root of n, or n for that matter? So the samples, what is the sample standard deviation over here again? We collected samples from 10 nest, and the standard deviation is written in the question itself, 24.
24.
Yeah. And what's the population standard deviation that we are trying to achieve?
4.
4. So 24 upon root n is equal to 4. That would give n somewhere is equal to 24 upon 4 is 6. 6 equal to root n. N is
6.
36. Alright, so now that we have done this, can we also see if we can do this? So I've reduced the range within which we want our 95%, within which the confidence interval should be 95%. So I've reduced that from 16 centimeter to 8 centimeter to see how many samples do we have to get now. Everything else is the same, I suppose. Yeah.
We see 144.
Anybody else? Yeah. I mean, yeah, Sachin, your answer is we are considering the answer. Let others also try. Is what I'm. So again, there's 8 centimeter range within which 95% confidence interval falls. So 8 centimeters again, 2 standard deviations from this side of the mean and 2 standard deviations on this side of the mean. So they feel basically that is 4 standard deviation within the 8 centimeter. So what is the population need to drop for an office call? Will join. Okay. Sure, Rusbeh. Yeah, so what is the population standard deviation that we are trying to achieve in this case? That is basically 8 by 4 standard deviation. That is, people interact a bit.
It's 2 centimeter.
Yes. And from our sample of just 10 nest, we got that our standard deviation we are achieving is 24. So if we apply the formula over here, the sample standard deviation upon root n equal to the population standard deviation, then that would mean 24 upon root n is equal to 2 centimeter. That would mean that n is equal to 12 into 12, 12 square basically. That is, the answer is 144. All right. So this is given in Suhail's lecture as well. If you, I mean, he has obviously explained in a much better verbose way. So I mean, this is me trying to explain the same thing. So yeah, if you haven't gone through Suhail's lecture, I think it's high time that you do before you go ahead to the other lectures, I suppose. Okay, so these tests hasn't exactly been explained in Suhail's lecture, but I thought these are a few of the simplest tests before you go ahead with the other complicated tests. So I thought we just have a glimpse of what all the tests are. So yeah, most of you have answered A. Can people explain, anyone who's who, yeah, can anyone explain like what was the reasoning behind picking A? Yes, Dev, you can go ahead.
Okay, see, basically, you're selecting the same bird before migration and after migration.
Yeah.
So, and comparing that difference as a data.
Yeah.
So there is a kind of pairing involved.
Yeah.
That's why I called it a paired sample
Sample
test.
test. All right. So that was nicely explained, and the answer is right. It is paired sample test. Let's see what we have in the next. Yes, Tarun. So birds could be different, could not be different. We don't know because they are all unmarked. But it's from the same population, that we know. So yeah, since we have so this is what usually happens on the field, it's very difficult to mark the birds and then track them during migration. That's like very difficult. So you usually have unmarked birds, and what people do is after and before migration, they can measure the weight of these birds, and so it definitely do not know that we are marking the or we are measuring the weights of the same birds, and so we're just measuring the weights of the population on this side and weights of the population on this side, and they're basically two samples. So and they're independent of, I mean, yeah, mostly independent of each other. So that would that would make us uh, do the statistics, I mean, make us do t-test on it, and it would not be paired t-test, so it would be basically independent sample. I mean, it's also called students t-test. I mean, if you want to correct me, please correct me, but students t-test is also very similar, so you can google that up and how it is done. There's a particular formula for it, and yeah. So the egg brooding period is basically where the egg is being brooded on, and the nestling period is where the eggs are hatched and the nestlings are out and demanding for food. And this is in a this is in a altricial chick nest, not the other one. Dimorphic is basically birds which where the males and females can be separately identified. Like a peafowl is a dimorphic bird where the peahens and peacocks look very different, starkly different. White ramshama is a dimorphic bird. Uh, yeah, a lot of birds are dimorphic. Yeah. Minivets, some of the minivets are dimorphic, quite 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 dimorphic. Okay. Uh, so paired sample t-test we understood when both the birds are when so basically paired sample t-tests work best when the individuals are the same between which we are trying to find a difference, and there's an after and a before. So paired sample t-test would be done best when we are looking at people trying a weight loss medicine or something like that. So you can do a weight. I mean, we can measure the weight before the ingestion of the medicine, and after the ingestion of the medicine for, I don't know, for a month or something like that. So so you have the same set of people, but you have a before data and an after data. So that's where the pair sample t-test works best. In our in our marked birds example, that was a paired sample t-test because there was a before weight of the bird and the after weight of the birds, and the birds were the same in both the cases. So, and we know, so bird number one has an after has a before data and an after data. Bird number two has a before data and after. So there's a paired sample in that sense. Independent sample t-test can be done if you know how to do it, but this is more of a chi-square t-test. Um, I'm not sure. I mean, are you guys? You guys must have been in some sort of. None of you guys are into data analysis or something like that in your respective fields.
Not.
I mean, I'm read. Yeah, Dev. Yeah, go ahead. Yes, Dev. I think you're on mute.
I didn't unmute it
Yeah.
myself. I thought chi-square test
Yeah.
is used to find a correlation kind of thing. See, when the chi-square has a certain value, the two variables are correlated or not. I'm not sure whether, see, here we are doing such a thing.
Okay.
And even, sorry to interrupt.
Yeah.
Even I think this chi-square test is mainly used to find out the causation correlation.
Right, causation or correlation, that's kind of
Yeah.
thing. That's what I thought also.
That's okay.
Yeah,
I mean,
one-
that's okay. Most of the
It is very similar to the correlation coefficient. I mean, not similar, but almost the same purpose, I thought.
yeah.
So
but,
we are not really finding any causation between the two comparison points,
but
frequencies of males and females.
no, chi-square can be. I mean, I don't think it is for correlation and causation. How what I learned was you can have two categories. You can have a male and a female, and a before so egg laying period, egg whatever brooding period and a nestling period. So there are two categories, and so that would make something like a matrix over here. So you have a male and female on the horizontal, and you have egg brooding and the nestling period on the vertical axis, and you have like one two. So it could be male in egg brooding, female in egg brooding, male in nestling, and female in nestling. So you have frequencies. So 4 frequencies you can have. Then you can see, yeah,
What were you having on the y-axis?
egg brooding and nestling. So 2 periods.
So two categories.
Yeah, 2 categories.
And on the x-axis, two categories as male and female.
Yes.
So you get 4 quadrants.
Yes.
And you are saying 4 quadrants and then do it with a chi-square. Okay,
Yeah, then we can do it with
brush up.
Yeah. So basically, what we do is I mean, ideally, if we our null hypothesis. I mean, this was in the last lecture where Suhel was talking about null hypothesis and alternate hypothesis. So ideally, what would be our null hypothesis? We will think that the male and females have equal responsibility, so they will come equally. So their frequency of visiting the nest would be equal. So suppose we have 100,000 samples of, or 1,000 samples of looking at the males visiting the nest and females visiting the nest. So if it's the null hypothesis, what we expect is in each of these stages, the males would have equal frequency. So that would mean that 250 males have visited in egg laying period, 250 visits of males in nestling period, 250 visits of males in nestling stage, and 250 visits of females in the nestling stage. Do you get my point till now? Yeah, I don't know what association studies. I have not done it. Probably that's what it's called in your field of study.
Yeah, this is still I am not really convinced.
So yeah, look up the chi-square. So there's an observed value, then an expected value. So what we generally do is observed minus expected upon expected square is somewhere. I don't know exactly remember the formula. I mean, it's very easy to do it on laptops and R nowadays, so you don't exactly have to know the formula. But once you do that, then you'll have a chi-square value, or you'll have something. So then it can tell you whether the male was visiting more in the egg-laying period or less in the egg-laying period, and whether the female was visiting more in the egg-laying period or less in
I think it would be paired sample t-test because the nest is the common thing there. So, and the nest in the egg brooding period, how many times male visits, how many times females visit. So we have two means there.
Okay.
So this can be compared with the paired sample t-test.
So t-test can be, in a lot of areas, t-test and chi-square can be done together in certain data. So you can go either way. But say that again, Astagimant,
Yeah.
how did you do it? Yeah,
I just repeat. So here the nest is the common thing.
sure.
Okay. So we have one egg brooding period. So during the egg brooding period, males visit for a certain times
Yeah,
and females visit for a certain time.
yeah,
When we have two means
yeah.
which belongs to same nest,
Huh.
I think paired sample t-test can be applied here.
But okay, because they're
If you are comparing between the males and females.
yeah, but I mean, I did tell you that we will have to sample. Yeah, yes, the nests are similar, but I did mention that we have to sample on the same individual and not the nest. Or
Same individual and same nest. In that case, it will be paired sample t-test. It will be male or female. So one nest, so one male and female. So let us say male visits 5 times, female visits 10 times.
okay, we're talking about
Okay.
frequencies.
Again, another nest is there. Again, there is a male. So male visits 6 times. Again, female visits 8 times.
Okay.
So we get a mean for the male and we get a mean for the female.
Oh, like that. But
At least I thought it is like that.
but it's not exactly an after and before effect.
It is not in after and before. That's why I said it is not exactly chi-square test.
Also, paired sample usually take into account the differences. Yeah, you can do it. So you can say the difference between the frequencies of visit between male and female. Yeah, that's an interesting way of looking at it. So what we generally do is you get if you're doing paired sample t-test, you get just one sample. I mean, you get the data is basically reduced into one set. So you'll have two sets, right? An
Yeah.
after and a before.
Yeah.
So
So one for the egg brooding period we compare males and females.
Yeah.
And another for the nestling period we compare males and females.
So you're comparing them separately.
Yeah.
Okay. So
So next, if you want to compare between the egg brooding period and nestling period,
Yeah.
again males you can have two data and you can compare,
Yeah.
and females also you have two means and you can
So
compare.
basically, what you're doing is you're doing 4 pair t-tests, right?
Two pair t-test.
2 pair.
One for the egg brooding period and one for the nestling period.
Sure, you can do the same
So-
with the male and female. So males in egg brooding
Yeah.
and males in nestling.
Again, if you are comparing for male and female, like it will be another two.
Oh, all right. Yeah, it can be done, but chi-square does it in one. But yeah. But you understood how to go about a chi-square test, right? I mean.
We know it generally in a disease. This thing causation, we use this
Chi-square test.
chi-square test.
How do you what kind of data would you have?
So, for example, hypothesis is smoking causes the cancer.
Okay.
Okay, we look at the how many people have the cancer, how many people don't have,
Okay.
and how many people were smoking, how many people were not smoking.
All right. Yeah.
So then it becomes a classical way of looking into. So you can say smoking is the cause for cancer or not.
All right. So you have smoking non-smokers and cancer non-cancer. Okay. Yeah.
Yeah.
So it's the same over here as well. Just that it's not exactly. I mean, I don't know. I mean, yeah. Okay. Sure.
Okay, let us think over it.
Yeah. Okay. Cool. That was a nice way of. I mean, yeah, but we when we were studying statistics, we did come across this where certain t-test and chi-square test can be done for the same data and come. I mean, the answer could be achieved in two different tests in two ways.
Generally, if we are establishing a causation,
Yeah.
like smoking causes the cancer,
Okay.
in such cases, we use the chi-square test, definitely.
Okay.
So here we are not really establishing any causation, like because male comes so many times, female is coming so many times.
Yeah.
Yeah, it is because of male is coming 5 times, female comes 10 times. No, it is not like that. We are not establishing any causation here.
But
So,
I'm not.
hence, I say it is. It may not be suitable. Chi-square test may not be suitable.
But I'm not sure if causation is the only thing used for. I mean, I'm really not sure. Huh? Probably we'll have to look it up. Actually,
Yeah.
I'm doing the same thing right now. Whether two categorical variables are independent in influencing the test statistics. When the test statistics is chi-square under the null hypothesis, okay. Yeah, it doesn't say exactly causation, but yeah, I mean, probably in your field it's more of a causation and correlation kind of a thing. But I think chi-square can be applied in a lot of other places as well, where you're not exactly looking at causation, you're just looking at two categories.
Chi-square can be applied, but which one is more suitable? Like that, we look at. If we look at, I feel it's a paired sample t-test.
So yeah, that's what I said. You can look at. I mean, obviously, paired sample t-test also works over here. You can have different answers, but chi-square also can work over here. Is something. Yeah. All right.
And just looking at the similar definition online, it is saying that, I mean, it was referring to this null hypothesis thing. So, are we looking at this problem from the perspective that we assume that male and female both will look at the test equally in egg brooding and the nesting periods?
Yeah. Yeah.
With that reference in mind, probably chi-square is applicable. But in case of and whether basically again the null hypothesis applies to paired sample kind of scenario also. That's what I think the question may arise later. So for the earlier example wherein we say that there are birds crossing the boundary and we want to check their mean weights before and after the migration. So were we considering that their weights will remain same, the mean will remain same?
Yeah. So we were considering, because in a paired sample, basically the differences between the before and after, the null hypothesis would be 0.
Yeah. So then does it mean that that time also it can be referred to as chi-square test?
I think it can be.
Can you do that with chi-square test? The weight? No, weight. Chi-square test is mostly done with frequencies. They are not done with such data. The weight of the animal or something like that.
Chi-square test is done with the frequency.
Frequencies. So there are two categories. Yeah. I mean, yeah. So not with the weight of animals. You cannot. That data cannot fit in chi-square. You need to have frequency data for that.
Okay.
Frequency as in how many in this category, how many in that category, and how many in the third and the fourth category. So that can, like.
Okay.
In that condition, we are just comparing the weights, mean weights. Then it is a t-test.
Okay.
Yeah, all right. So basically, this means that we have a lot of reading to do. So we'll just go ahead.
Yeah, let's go ahead.
Yeah, I did. Chi-square formula.
No, I searched in Google and found it, but I'm just having it with. So
Sure.
don't bother.
Okay. So yeah, over here we have we have three categories instead of the usual two categories that we've been having till now. So over here we have males, females, and juveniles. Three categories. We have data of song lengths for males, data of song lengths for females, data of song lengths for juveniles. Definitely it can't be paired because all the three are different individuals. There's no after before. And yeah, but we could still carry out t-test if we have to measure like males and females. uh, if you only consider males and females, then we can have t-test between males and females. We can test between females and juveniles, again with a t-test, and we can do the same with males and juveniles again with a t-test. So yes, we can do t-test as well, but since we have three and we want to see is there a difference between the three, the best uh, option over here would be ANOVA, just like a short form for analysis of variance or something. um, so have people over here done ANOVA in some situations? Anywhere? Yes, Dave. Oh, so you have done, you're saying. All right. If you
We
want.
have also done that one.
Yeah,
All right.
basically studied it. Okay, fine.
Okay. So, so, so when you do ANOVA, can you can you point the exact population which is like way different from the others, like exact, like if we are doing males, females, and juveniles, and we do ANOVA and we find that the ANOVA test is significantly different. Like it says that one of the categories is significantly different from the others, or one or two or whatever. Can you tell which category would be different from the ANOVA test? You understood my question, right? Okay, so we are looking at
Purpose of ANOVA test is to compare the changes or the data variance or something for
Yeah,
the samples, right?
for more
Of
than
different categories,
yeah,
correct?
for more than 2 categories.
So it can be easily found out whether a
T-test.
particular group is different or not, or how they are correlated, right?
Yes, it's the same thing. We can do it with t-test. It's just that if there are 3 or 4 or 5 categories, sometimes you can have them, and you want to figure out if any of the categories or more than one of the categories is actually different from the others. So over here, what could be the hypothesis? Like, let's just figure out, like, there's a male song length, there's a female song length, and there's a juvenile song length. What could be the hypothesis over here based on song length?
Male, female will have a shorter song length. That's what I would assume.
You think juveniles would have a longer song length?
Shorter.
Juveniles would have a shorter, and male female
Yeah.
would have a longer.
They have same. That's what I would assume.
All right. And some cases, even males would have much higher, and females would have still less than males.
Probably, yeah.
Yeah. So that is our hypothesis. So basically, what we are thinking is, in all these cases, there would be a situation where males have higher. If we are doing a box plot of the song lengths, we'll have males higher, females somewhere over here, and juveniles the least, right? Yes, go with.
There are there cases in which there might be differences between the song lengths in individual birds?
There would be, of course, there would be. But that is what ANOVA looks at. ANOVA looks at the differences within the individuals of the population and differences between the differences between the individual. Sorry, what was it again? Within and within the category and between the categories. So if the differences between the categories are much higher than what is within the categories, then ANOVA test comes out, significantly different. If not, then basically all the categories have similar differences, so there is no difference between the categories. So anyways, this requires a lot of reading, and obviously this course also hasn't gotten any lecture on these kind of tests that you do, but obviously this is something that is important. And after listening to Suhel's lecture, probably you should go and look for studies that have that are on birds and that have used, and there would be very old studies. Newer studies don't exactly use these tests anymore, but you should go and look for older studies on birds that have looked at t-test and ANOVA test and see what kind of data they have used.
So
What is the latest replacement test, sir?
What?
What tests have replaced these older tests?
Now people, it's the same as regression analysis and stuff like that. People use a lot of other. It has a very similar context. I don't know how to explain that, but it's got a very similar way of finding the same thing. But nowadays they use regression analysis, and even in that there are like several other generalized linear modeling. Then you have generalized linear modeling with with effects like random and so a lot of these are the things that are people basically GLM, LM, LMER, GLMER. These are the names of the tests that people usually do, but they are all based on these but the basic ones that I'm showing you on the screen. They're all based on t-test, chi-squares, and ANOVA and all that. So yeah, Sachin, you were saying something.
I was just asking if there is an article which describes these different types of tests with some relevant example that I can refer to, or shall I just Google and check that thing?
Google, but that book would have, no? We did discuss about statistics for biologists, statistics for ecologists. There are a lot of these books that are available, so you can start with that.
we discuss in the lecture or today early because
Not
I
today.
joined.
Not today. Like a
Okay,
yeah, one of
okay.
the future, one of the
I
past
think
lectures.
you can look up that extra reading material which is given
Sure.
every
Yes,
week.
yes. I didn't check that. Yes, you should actually.
Yeah, I didn't check. Yeah, sure. I'll check that thing. Thanks, Mr. Dev.
Let me check if it's got. Uh, um. Yeah, probably you won't find these kind of tests in the extra reading material. You'll have to do a little more search on yourself, probably.
Sure.
I,
Thank you.
yeah. All right. So the next. So basically, so ANOVA would tell if there's a difference between the categories, but which two categories are different? That would basically be done. Anyone knows what is used to? Okay, I'll just. It's called a two-key test. Suppose there's so we find out we from ANOVA we find out that between males, females, and juveniles there is one category which is very different. Post hoc, yes. So to further find out if what category was really different, we do a two-key test and it'll tell you like all the combinations and which combinations actually basically do t-test amongst the combination and say which categories are really different, like significantly different from each other. Anyways, so we're touching upon the disease lecture of of Faraj now. Thank you, Tarun. Post hoc. Yes, that's the term that I was looking for. Yes, Dave, it could be a multiple choice question. The question says, what are the factors?
So I could give answers like A and C or something.
Yeah.
Okay.
Sir, on some level, all four options can apply, right, sir?
Just tell me the options then. Okay, I'll not take much of your time over here. So the answer is basically all the four. On some level, yes, Govind, you are right. On some level, all the four. There are more options. I mean, there are more factors as well. I'll have to check again. I mean, desertification, urbanization, climate change, deforestation, human population is one of the options. The overpopulation of humans. Then, yeah. So there are furthermore, yeah, A, B, C. C is also possible. So enemy release hypothesis is basically a very famous hypothesis in invasion ecology. Invaders over here are invasive species encounter in an introduced range. So when I say invaders, those are the invasive species that have been introduced in a newer in an uninvaded area types. you can check on whichever you think has the nearest, or you think has the nearest meaning to what the hypothesis could possibly mean.
Sir, I've always been thinking about this hypothesis. Isn't it possible that the reverse might also be true?
Yeah, it is.
The introduced species could encounter pathogens it does not have resistance to. It
You
could
know-
work both ways.
Yeah, it can work both ways. It's just a hypothesis, like we say. So when we are studying, like I can share you a paper, like when we are studying these things. So not all the hypotheses are universally true. Some of them work for some setting, some ecological setting, like yeah, and some of them don't. There are other hypotheses that work over here. Some of them are partially true, partially work and partially don't. So yeah, opposite can be true as well. Yes.
Well, in this case, the hypothesis works best in a closed setting like an island, right, sir? In an island, the diversity and abundance of pathogens would be relatively low compared to, say, a large-scale open landscape. So the enemy release hypothesis would fit the best in an island, right, sir? Or a similarly small enclosed environment.
Possibly, yes. So islands have islands have a different dynamics with invasive species because they were historically historically distant from all that was happening in the mainland usually. So usually mainland have a very like already a lot of species have has gone from hands to hands and interchange in so a lot of things, so a lot of resistance, a lot of interaction between the host and parasites have already happened in mainland. So whenever we're talking about invasive, but yeah, for example, island need not be some island within an ocean. It can also be an island, something like a Shola Sky Island, which has a very different kind of a climatic realm, and it forms like an island of a very different species set, a different climatic variables. So that can also be an island in some sense. So
I know, sir. I meant like a closed habitat. Island is an example.
yeah, okay.
For those,
A cave would also be a good example, right, sir?
Yes, yes, yeah, possibly. Yes, Sachin, you were saying something?
I was saying, does this relation or the instance hold true for the what do we call some non-native species brought from somewhere else to a native land, like for example maybe bajrikars or some outside species brought to a native land, and will that also fall under this category of invasive species to the native?
So invasive species have they have the definition of invasive species has changed a lot, but so basically there's a non-native species first of all.
Right.
Non-native
Right.
species is basically as the definition says, they are not native to the introduced
Correct.
area, right? So they have been brought from a different country or a different biogeographical region and brought to this area or whatever, and hence it becomes non-native. So now non-native, how they react to the climate or to the situation over here can have 2 or 3 pathways. First they can have no reaction at all. They couldn't adapt, they couldn't bear the climate, and they just die. So that could be one reaction. The other reaction is they adapt, but they don't have very fruitful. Basically, they don't thrive very much.
Right, yeah.
Yeah,
right.
they don't thrive. Then there's another one where the climate of the non-native species was very similar to where it was introduced. So there, what can happen was it produces a lot of seeds, a lot of
Right.
offspring, and
Spreads.
it spreads. So then it becomes an invasive species. Till then it was
Oh.
just a non-native species.
Okay. I don't know whether it's correct or not. The African tulip that we see everywhere, will that be a
Which one is the
invasive?
African tulip? The Spathodia campanulata. Spathodia.
The orange colored flower.
Orange color that
right?
is there in
Right.
Bangalore and all. No, I
Yeah.
don't think it's a invasive species. It was a very beautiful looking species, and people have planted it at a lot of avenues. That's why you may find that there are a lot to see, but probably I don't think they're invasive species. I've not come across. I've physically never seen it invading. Like I have not seen seedlings or something growing out of the range of that plant, or I have not read anywhere that African tulip is an invasive species.
What I'm trying to,
Have you seen like seedlings growing elsewhere, like far away from the parent?
I do not know.
Yeah.
But
Also,
what
there
I'm
are,
trying to understand is basically when, I mean, I might have been wrong to understand this concept, but when a non-native species comes here and thrives and spreads a lot,
yeah.
isn't that kind of an invasion to the local species occupying the land and maybe the resources part I'm saying?
Yes, but we
basically.
are also talking about an urban area where Spathodia was particularly a very coveted kind of a species for avenue trees.
Right.
So it was the humans who actually planted them.
Exactly. I mean, humans, I'm saying, are not generically coming, right? I mean, on its own,
No,
like
no,
the birds.
that's what they have planted them. So what you see is that because they are
Right.
planted in on avenue trees where
Right.
visibility of people are very high, so you seem to or probably you seem to feel that they are spreading. I mean, they have spread a lot, but I
Okay.
don't think in a wild area you'll find a Spathodia sapling or a seedling coming.
So you're saying that if the species grows on its own,
Then
spreads it on its own, then
Yes.
it may be invasive.
that is where it is invasive.
I
If
see.
a human
Okay.
is helping it out, or if yeah.
True.
Also, what humans do can be controlled, but suppose, for example, lantana. Now, lantana has this property, like the fruits are very coveted by the birds, the local birds over here. So they eat the fruit and then they spread the seeds as well. So there's a vector for their spread as well. So even the lantana can spread through the roots as well, but it can also spread through the seeds now. So different ways of propagation for lantana. Lantana is known to be like a highly invasive species. So there's but again humans can't control both the spread of lantana or the spread of the birds feeding on lantana and spreading the seeds. Like it's very difficult. So again it becomes invasive in that sense.
Okay. And does that also happen with the Santa Maria feverfew, the Congress grass, what we call, I don't know.
Yeah. I know.
Okay.
Parthenium.
Parthenium, right? Yeah, Parthenium. Is this also invasive then?
Yeah, it is invasive. Yeah.
Okay.
It
Thanks.
has come from I think
Wheat, I think.
yeah, come with wheat and it has come from Europe, because if you see like some very old I don't know if you see some old movies from Britain, you will see Parthenium in the background. I mean, I've noticed this and I was really surprised. Like, oh,
Okay.
I see Parthenium in the Britain, like UK buildings. Sorry, UK movies. So you can see. So there's another species called gorse, which is very common in Nilgiris and Palani Annamalai Hills. Again, a very cold adapted species, but it's a very invasive species in Nilgiris. Again, you can see them very common in one of those period movies like I don't know, Pride and Prejudice or something. Some movies like that, I've seen them in the background. So I mean, it's obviously movie has been made in UK. So so those species, yeah.
Thanks.
Yes, Govind, and there was another person who raised his hand. Govind, before you
yes, sir.
can the other person take the question? You've already asked one question. Can Astagmit, sorry.
Sure, sir.
Did you have a question, Astagimat?
No, I was giving the example of lantana only. You just took it.
All right.
Yeah.
Yes, Govind, go ahead now.
Yes, sir. My question is there. It's actually a piece of information I once read, saw in a documentary that invasive species require some kind of an ecological foothold or a foot in the door in order to actually become full on completely invasive. Like apparently Asian carp is a very invasive species in the North American rivers like the Mississippi, and they have colonized very large stretches of the river which come under human influence. But parts of the river which are still healthy and going strong, they haven't been colonized to the same extent. So apparently, invasive species, at least some invasive species, might need some kind of an opening to exploit. They can't become invasive immediately.
Yeah. I mean, there are a lot of theories now coming up these days. Like people have come up with the theory that invasive species usually do not invade where the climate that they have been facing in their original habitat is very different from the introduced range. So there, those invasive species don't do very well. So those kind of theories, like a lot of theories have come up. So yeah, I mean, invasion ecology is like booming right now. Anyways, this was more about the disease ecology. So enemy release hypothesis is basically invaders encounter reduced pathogen abundance and diversity in introduced areas, and that's why they have a greater like Govind said, they have this foothold, they have a greater advantage over other species where the pathogens are already acting upon. So yeah, so that is the enemy release hypothesis. Let's go to the other question, which says the novel weapons hypothesis. Yes, Govind.
Sir, could you say that invasive species is a factor driven mostly, is a factor that came into existence after humanity, or do you think it's been in existence for far longer than that?
Yeah, that's a very good question, but I'm not sure how to answer that. Like, where do you draw a line? That's the problem with invasion ecology. Like, it's mostly seen from a very human centric perspective, um, and yeah, so where do you draw a line is something that, yeah, because
Well,
if we consider when the humans were not there, or even before that, when the earth was entirely a Pangea, like one piece of land, all of, I mean, it was one piece of land. So we often use like, oh, this was this was native to India and not to, I don't know, Europe or whatever. So if we consider the entire thing as one piece of land, then I mean, cannot have invasive species. And I mean, it's already all connected. It's like one mainland where things were already exchanged. So, yeah, I mean, a very tricky situation there. But usually, invasive species, I mean, are considered after the arrival of humans in the sea. Like sometimes even human beings have been invasive.
Yes. Yes, sir. Well, even before human beings came to existence, like tens of millions of years ago, I have heard of stories where different species travel to newer continents or newer islands through the process of rafting or land bridges. Could they also be considered invasive species to some extent? And it's before human beings' time.
The only difference I think is the intensity of this exchange of biotic material or something like that. Yeah, it's the exchange, like how frequently is the exchange happening. So it can happen that a migratory bird brings one parasite from a long distance away uh, uh, to the south, but the frequency won't be as high as how humans have carried out this exchange of biotic materials from one continent to the other. So probably that can answer a lot of questions in that sense. Like, where do you draw a line?
Yes, sir. So you
Okay.
could also say that this has happened in human history as well. I mean, humans have naturally introduced diseases to different other communities of people across the world.
Yeah. Yeah. I mean, of course, if we look at the history of America, where the Europeans brought what was the disease? Smallpox to the native Indians. So they gave the smallpox ridden blankets to the native Indians. I mean, thinking that, I don't know, but they so the blankets were given, I don't know for what reason, but they actually gave the native Indians who were not used to the smallpox virus. They were not vaccinated at that time, so they got the smallpox virus. They died. Their population almost decimated because smallpox is not a small disease or something. So I mean, that is also something like an invasion in that sense. But anyways, let's not, that is a very political thing to discuss. But yeah, so novel weapons hypothesis, basically invaders carry new pathogens that spill over to the competitors in introduced areas. So basically invasive species are bringing newer diseases into the introduced areas, killing the other native species, and then finding a foothold in the new area. So that is a novel weapons hypothesis. Yeah. So this was a last minute question, so please forgive me for the crudity of this question, but yeah. If you don't want to give answer for C and D, it should be okay. Which is true amongst A and B is something that I'm looking at. Between A and B. So Hemoproteus and Plasmodium are two taxa of these pathogens that are usually found in birds, and one of them is a host specific pathogen and the other is a host generalist pathogen. So if you can answer me with A and B, which is which, that should be okay.
They're both carried by mosquitoes, right, sir?
Yes, and other things as well. Like one of my colleagues is working on that, and he says it's not just mosquitoes. There are something called midges, MIDGES. They also carry one of them, and then yeah, those are the only things that I know. But yeah, there are other species as well, other vectors as well.
I remember once reading that Plasmodium might be relative of algae, right, sir?
Relative of algae, yeah. I'm not sure. I'm not sure about that.
I think I saw it in a video somewhere. They found very trace amounts of chloroplast organelle in Plasmodium, and which is related, which explains that it was it comes from a similar family to algae.
Yeah, possible, very possible. I mean, microbial worlds. I mean, that's why I never got into molecular biology. It's too too complicated for my tiny brain to understand. But anyways, yeah, the answer is there on the screen, A. But if you're looking at C and D as well, the host-specific pathogens are usually more would be attracted to show like yeah, I mean that was a very it was a last minute question, so please forgive me. I mean, A is definitely answer and the answer. Hemoprotease is known for its host specificity, and Plasmodium is known for how generalist it is. Like hemoprotease would be specific to certain species, and Plasmodium is not very specific to any species. It would be a very host generalist species. So both of them have different properties that way. So yeah, and in the studies in the disease ecology studies, it's usually said that homoprotease is host specific. So the studies are designed according to that. Plasmodium being host generalist, studies are designed according to that. So yeah. Anyways, this was the last question, and we'll end the session over here. Yes, go on. Astagamet, please carry on. What are you trying to say?
My doubt was, so in my assignments,
Yeah.
one of the assignments, there is some discrepancy, so I should contact whom? Just I wanted to know, is there anything you can tell?
The last day for the assignment was yesterday, right?
No, not the sixth assignment.
Oh, sixth assignment. Oh, that's like okay. That would have to be the NPTEL coordinator or yeah.
Is there any particular mail ID?
I can.
Where can I find it at least?
In your
write in support forum, no?
Yeah.
Write a mail in the support forum.
Okay.
Yeah,
Okay.
so the NPTEL people, immediately take care of that. Yes.
Yeah. Thank you.
All right. Anything else?
Oh, thank you.
Sure. Then can we disperse? Can we call an end to the session today?
Yeah, it was fine. It was a very good discussion.
All right. Good night then. Bye.
Good night. Thanks.
All right.